Hello, everyone, and welcome to our presentation. So, today we are going to talk about the significance of MUC2 gene methylation detection in pancreatic cancer diagnosis. So, this study was performed by Zoe and his associates back in 2019, and the presentation will be done by me, Jessa, and my partner, Juveline.
Hi everyone, this is Juvelin. To start off our presentation, I want to give you guys a little background about pancreatic cancer. So, pancreatic cancer ranks as the eighth most common cause of cancer death worldwide, with an early metastasis, meaning that the cancer spreads to different parts of the body from the initial site. It also has a high degree of malignancy, meaning that the cancer cell grows and spreads rapidly throughout the body. It is also known that pancreatic cancer has a very poor prognosis. Due to that poor prognosis, a person with pancreatic cancer has less than 9% of survival rate. The aim of this research is to investigate the diagnostic quality of MUC2 gene methylation in pancreatic cancer and compare that off to the present diagnostic method called CA99 assay. Welcome to Materials and Methods section. The cell culture that was used in the experiment was AD293 human embryonic kidney cells and were purchased from Shanghai Science and Biotech Corporation Limited. The pancreatic cancer cell lines that was used in the experiment were SW1990, ASPC, BXPC3, CFPAC, PANC1, PATU8988, and were purchased from ATCC. All of the cells were submerged in DMEM with fetal bovine serum, penicillin, and tryptomycin, and they were incubated at 37 degrees Celsius, 5% carbon dioxide, and saturated humidity. Blood and fecal specimens were also collected. Blood were collected from 40 patients with pancreatic cancer, 15 patients with chronic pancreatitis, and 25 healthy patients to act as a control. Fecal specimens were collected from 30 patients with pancreatic cancer, 8 patients with chronic pancreatitis, and 20 healthy patients to act as a control. So the first step of the experiment is to extract the DNA from the pancreatic cancer cell lines, fecal samples, and blood samples prior to methylation. DNA extraction is a process used to free the DNA from the cell and then separate it from its cellular fluids and proteins so that you are left with a pure DNA. The process of DNA extraction consisted of cell lysis, precipitation of cell content, and purification of DNA. So after extracting the DNA, we can now proceed to the second step of the experiment, where the DNA fragments from the three samples were then digested with a restriction endonuclease, HIND3 and methylation restriction enzyme, MSP1, and HAP2. So restriction enzyme is a protein that recognizes a specific sequence within the DNA and cleaves or cut the DNA only at that specific site known as restriction site. The result on MUC gene methylation in blood specimen showed that patient with pancreatic cancer has 40% methylation rate, which means that only 16 sample methylated out of the 40 samples. Meanwhile, patients with chronic pancreatitis showed 0% methylation rate, and healthy patients, which acted as a control, showed 4% of methylation rate, which means only one methylated out of the 25 sample. MUC2 methylation rate in pancreatic cancer is significantly higher compared to the chronic pancreatitis and the control. In the blood sample, MUC gene methylation displayed higher specificity of 97.5% and positive predictive value of 94.1% compared to the current diagnostic method CA19-9. So moving on to the result on MUC2 gene methylation in fecal specimen, it showed that patient with pancreatic cancer has 43.3% methylation rate, which means that only 13 sample methylated out of the 30 samples. Meanwhile, patient with chronic pancreatitis showed 0% of methylation rate, meaning that none of the sample methylated. And healthy patients' control showed 5% methylation rate, which means that only one methylated out of the 20 samples. Once again, MUC2 methylation rate in pancreatic cancer is significantly higher compared to chronic pancreatitis and the control. In the fecal sample, MUC2 gene methylation displayed higher specificity of 96.4% and positive predictive value of 91.7%. And once again, it is compared to the diagnostic method CA199. So the overall result of the experiment statistically support that there is a significant MUC2 gene methylation detected in pancreatic cancer patients in comparison to patients with chronic pancreatitis and control patients, particularly in the combined blood and fecal specimens, as it shows a hypermethylation in 6 out of 10 pancreatic cancer patients, showing a statistically significant difference between the hypermethylation in pancreatic cancer patients versus chronic pancreatitis and the control patients, which showed a probability of less than 0.01, which means that the observed difference is not by random chance. And compared to the CA199 assay, diagnostic specificity and positive predictive value were improved by the detection of the MUC2 gene methylation in the peripheral blood sample for the pancreatic cancer diagnosis. However, this promising diagnostic tool is still at its infancy, meaning it's still at its early stages, which means it requires a more controlled clinical trial to be thoroughly studied. And that's the end of our presentation. And we hope that you guys learned something out of this research study. And thank you for listening and have a great day.